Hello, everyone, and welcome to today's live broadcast, CAR T-cell immunotherapy of solid tumors, learning from the clinic and lab in parallel. I'm Alexis Krause of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Lumix. For more information about our sponsor, please visit their website at lumix.com. Now, let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. I'd like to now introduce our presenters, Dr. John Maher, clinical immunologist, scientific founder, and chief scientific officer at Leucid Bio, and Dr. Rogier Reijmers, principal scientist at Lumix. For the complete biographies on our presenters, please visit the Biography tab at the top of your screen. Dr. Reijmers, you may now begin your presentation.
Hi everyone, welcome to this joint webinar. My name is Rogier Reijmers and I'm the principal scientist of Lumix. And my part will cover how using Lumix Zimovi can help in selecting the best CAR lead candidates for immunotherapy by determining an additional important parameter, that is cellular avidity, which reflects the binding strength between T cells and target cells. But first, shortly, I would like to introduce Lumix. Lumix was founded in 2014 and has its headquarters stationed in Amsterdam, the Netherlands. Over the last 6 years, we've grown from a small startup company with only 2 employees to a scale-up that is currently joined by almost 120 gifted people from all over the world. Lumix develops instruments for scientific research, and in April of this year, we launched our newest product, the Zimovi Cell Avidity Analyzer, which is available for sale. The Zimovi quantifies cellular binding avidity directly and in a high throughput manner at a single cell level. And in the second part of this webinar, we will show that avidity has predictive value for CAR T cell functionality. That Zimovi avidity measurements are reproducible because avidity is measured using a biophysical method. And it's also that Zimovi is fast. You can obtain your avidity results of hundreds of interacting cell pairs within minutes. And of course, but why did we develop this technology? Why are these features important? And we think that it can accelerate the identification of lead candidates. As great successes have been achieved, adoptive cell therapy still has its limitations and drawbacks. Most importantly, optimal preclinical design, including CAR engineering, are a work in progress and still require refinements. In many patients, treatment with CAR T cells, responders develop cytokine release syndrome, often in combination with neurotoxicity, which, when left untreated, can be fatal to the patient. And another major issue is the fact that CAR T cells mostly fail when applied to solid tumors. However, when targeting tumors with less specific markers, unlike B-cell malignancies, there is a high likelihood of on-target of tumor toxicity, which could be fatal to patients as well. As a result, lead candidates could fail clinical trials. For this reason, many clever approaches have been developed by applying complex engineering to CAR expressing T cells that could result in more specific therapies with reduced off-tumor toxicity, but also minimizing T-cell exhaustion. These include, for example, targeted insertion of a CAR into the TCR locus, which leads to a more physiological expression pattern and reduced exhaustion, or making use of dual CARs that recognize 2 targets, which is required for activation, thereby reducing off-tumor toxicity. And finally, a strategy that could lead to reduced off-tumor effects is affinity tuning of existing CARs that otherwise easily recognize healthy tissue expressing low levels of tumor targets, or increase the efficacy of CAR T cells, something as was recently shown for FMC63, a well-known CD19 CAR. Here, two CARs, FM63 and CAT, which has been derived from FMC63, were compared. They both target the same epitope of CD19, but with a very important difference. The affinity for CD19 of CAT is 40-fold lower. What you can see here is very interesting, as the low affinity CAT CAR performs better in terms of tumor cell killing in vitro at every E/T ratio explored in this study. When the same two CARs were compared in an in vivo preclinical mouse model, you can see a similar pattern where the high affinity CAR is only capable of slowing down the tumor compared to the control, but the low affinity CAT CAR can reduce the tumor burden very significantly. These data show that having a high affinity CAR will not per se result in the best T cell function. So a fair question to ask is, do we have the right tools in place to efficiently select the best lead candidates? In essence, the traditional biophysical methods primarily focus on affinity for CAR lead selection, generally known as the strength of interaction between one ligand and one receptor. This type of interaction completely neglects the biological complexity that is inherently linked to cell function, and therefore what is missing is the cellular avidity that can be described as the overall strength of interactions between a diversity of receptor ligand pairs at the cell-cell interface, as shown in this figure below for a CAR T cell interacting with its tumor target. Not only is the CAR binding to the target, also many other interactions must be considered, which, for example, includes adhesion or co-stimulatory molecules. To be able to measure such interactions, there is a need for technology that allows direct, fast, and high throughput measurements of T cell binding avidity. And for this reason, we have developed the Zimovi cell avidity analyzer at Lumix. Because it is becoming more and more clear that the anti-tumor response is really driven by the overall collection of interactions that take place between the T cell and the tumor cell, it is the T cell binding avidity that drives tumor killing through the formation of a strong and mature immunological synapse. In this specific synapse state, the interaction forces substantially increase because of the active recruitment of many adhesion molecules at the cell-cell interface. And only in this state, the T cells can execute the killing by very locally delivering cytotoxic molecules. Zimovi can measure the binding strength at the most critical stage of this process, where the synapse is forming. How does Zimovi measure this cellular avidity? The Zimovi relies on a small microfluidic lab on a chip shown at the top of this slide. Target cells are seated on the bottom of the glass surface, and T cells are co-cultured on the top of the monolayer. The flow cell is 100 micrometers in height, and the piezo element in yellow on top can generate standing waves in the flow cell medium. These standing waves can be compared to sound waves coming from your radio speaker. If the magnitude of the force exceeds the binding strength of the cell pairs, the T cells will be lifted into an acoustic node, which can be considered a zero gravity area, where cells float until the piezo element is switched off. In this way, Zimovi makes use of acoustic forces to separate cells, and the magnitude of force that is needed to separate these interacting cells is a direct readout for their avidity. Let me show you a movie of how this looks like when you run a real experiment. Here you see fluorescently dyed T cells on a monolayer in black. When you start the acoustic force, you can see that the cells accumulate in the acoustic nodes. And in this field of view, you can measure the avidity of up to 300 to 400 cells in one go. So how do you get to a run like this? Here you see a simplified overview of the workflow. So let's go a bit into more detail. First, you select your target cells, which could be a panel of cells expressing different levels of antigen. Typically, one uses 100 million cells per ml in a maximum volume of 250 microliters, which is needed for 1 run, with a maximum of 5 runs per reusable chip. Then you flush in the target cells and seed the chip to generate a monolayer. The incubation time on average is about 2 hours. Preparation time for an avidity assay will be no longer than 30 minutes. Then you prepare your control or engineered T cells and dye them with a red fluorescent dye for tracking, as shown in the movie on the previous slide. T cells are used at a concentration of 10 million cells per ml, and you need only 30 microliters per run. Preparation time for an avidity assay generally is about 1 hour. And finally, you incubate the T cells with target cells, which takes about 5 minutes, which is the time that they are allowed to form a synapse. But this is flexible. You can start the acoustic force ramp, which is standardized to a 2.5-minute force ramp going from 0 to 1,000 piconewton, but this is flexible as well. You could also use a static force or go even higher, although that could affect the quality of the monolayer. After the force ramp, you wash away the T cells and incubate your next T cell population, up to 5 runs. Altogether, when incubation time is set to 5 minutes, 5 sequential runs will take about 1 hour of experimental time. This will lead in the following figures. Here you see an example of avidity curves that can be obtained after an experiment. The curves depict the percentage of bound T cells to the monolayer as a function of force. Based on a negative control, a background binding gate can be set, which usually coincides with the moment the negative control cells no longer display a detachment event. This gate determines the minimal force that is required to release all non-binding T cells. This gate then determines the percentage of T cells that specifically bind to the target monolayer, which can be shown in a bar graph. It reflects the percentage within your T cell pool that specifically interact with the target monolayer compared to the negative control, and these T cells require an additional increase in force to break the specific bonds. One has to realize that the avidity curve consists of individual events of single T cells that detach from the monolayer, each having their own specific force. This can be plotted as a violin dot plot. These are the type of graphs that we use to show our data generated with the ZMOVI. As proof of principle, I'm showing you a data set that was collected by Professor Stephen Abelda, one of our collaborators at UPenn, who is doing research on CAR T cells targeting lung cancer. What you see are 2 CARs that target the same antigen, but they revealed different levels of in vitro cytotoxicity, either high or low. In the presence of high antigen expressing tumor cells, you immediately see that the avidity curve of the high functioning CAR in red is higher compared to the low functioning CAR in yellow, which both have a much higher avidity than the background controls in black. When you zoom in at the specific acoustic force of 250 piconewton, which is the background gate, you see that the high-functioning CAR T cell population has a far greater percentage of specific binders than the low-functioning CAR. One may ask whether this behavior is dependent on the presence of the specific antigen and what might happen when the antigen is not expressed by the target cells. As such, when measuring avidity in the absence of the target antigen on the monolayer, none of the T cell populations bind. T cell binding avidity is very low in all 3 cases tested, and only background binding is observed. This technology has also been used by our next presenter, Dr. John Maher. He will explain how he identified the most potent CAR amongst his affinity-tuned panel using ZMOVI, which resulted in the best performing CAR in a novel configuration that he developed, known as parallel CAR T cells. I'm excited to hear about the latest development, and I'm looking forward to his presentation. Please, John, the virtual floor is yours.
Well, hello everybody. My name is John Maher. I'm the Chief Scientific Officer of Leucid Bio, and it's a pleasure to present at this webinar hosted by Lumix today. I guess my presentation today is going to be the proverbial game of 2 halves. Initially, I'm going to tell you a little bit about a clinical trial that we have running involving a CAR T product here in London at Guy's Hospital. And then I'll describe some preclinical research that we have been conducting, developing a next generation CAR technology, which we refer to as a parallel CAR. And I'll tell you a little bit about how we use Lumix Z-movie technology to enable us to select targeting moieties or binders, which proved useful in this work. And this slide shows my disclosures. So the clinical trial that I'd like to tell you about involves a product that we refer to as T4 immunotherapy. And T4 immunotherapy involves the co-expression of two chimeric receptors in patient-derived T cells. The first of these is a second generation CAR shown on the top of this cartoon, which we refer to as Ti28 zeta. So the signaling components within Ti28 zeta are by now pretty standard. It's a fusion of CD28 with CD3 zeta providing signal 1 and signal 2 to activate these T cells. What's a little unusual about this CAR is the targeting moiety, which is not an antibody fragment, but instead is a chimeric polypeptide known as T1E. And the T1E polypeptide is in fact a promiscuous ERB-B ligand. It can engage 8 different ERB-B homo or heterodimers and therefore provides very broad targeting specificity for epithelial malignancies. Now we're co-expressing Ti28 zeta with a chimeric cytokine receptor that we developed many years ago called 4-alpha beta. And this essentially harnesses the very poorly mitogenic cytokine interleukin-4 to deliver a selective, potent growth signal only to the CAR T cells. So it provides a convenient system to enable us to selectively expand our transduced T cells during our 2-week manufacturing process. Now, whilst targeting the extended ERB-B network offers a lot of potential in terms of treating solid tumors, it obviously also confers a potential safety risk because, of course, these dimers are also expressed at lower levels in many healthy tissues throughout the body. And it was for this reason that we selected head and neck squamous cell carcinoma as our target disease, because we viewed this as a disease model in which we could evaluate the safety and potential efficacy of intratumoral delivery of CAR T cells. The main clinical problem that these patients encounter is shown by this rather graphic image here. In other words, locally advanced or locally recurrent tumor formation. We also selected head and neck cancer because this is a tumor which highly expresses the target. These tumors are virtually ubiquitously positive for EGFR, and tumor cells generally co-express at least one of the other ERB-B family members. It's a cancer against which we haven't really made a great deal of progress in terms of 5-year survival in recent years, and it's still stuck at more or less 50% in these patients. Another key reason that we selected this malignancy relates to the prognosis for patients once their clinician explains to them that there is no suitable conventional treatment for their disease. This Kaplan-Meier curve was generated from data generated at our cancer center at Guy's Hospital. And what you see is that these untreatable patients begin to die of their disease literally from day 1. And by 30 weeks after receiving such a prognosis, all of these patients will have died of their disease. So for this reason, we viewed this clinical scenario as a kind of an ethically justifiable area for the delivery of this potentially toxic intratumoral CAR T cell approach. The design of our clinical trial is relatively conventional by phase 1 or first-in-man new cancer medicine clinical studies. So it's a dose escalation 3+3 study in which the first cohort of patients receive a dose of 10 million CAR T cells, which are injected as a fresh product at a single setting, injecting multiple points around the viable periphery of the target lesion. and we escalate through 5 different dose levels up to a maximum deliverable dose of 1 billion CAR T cells. More recently, we have opened a 6th cohort in which we have reduced the dose of CAR T cells to 10 to the 8 cells, but these CAR T cells are administered after lymphodepleting chemotherapy with the combination of fludarabine and cyclophosphamide. Being a first-in-man study, it's primarily about safety, but we have a number of secondary endpoints, translational endpoints, which are designed to inform us about the mechanisms and the persistence of these T cells at the site of injection. This is a rather busy slide which shows the characteristics of the 18 patients who have been recruited to date to this clinical study. You will see that 2 of the patients were not treated. Patient number 5 unfortunately died of his disease during the 2-week period that we were manufacturing the CAR T cells. And we had another patient, patient number 12, who by the time he presented to our center had vascular invasive disease, which is an exclusion criterion, and therefore we were unable to treat that patient. All of the other 16 patients were treated with CAR T cells at the target dose and without any manufacturing failures over the course of the study to date. Manufacture of T4 immunotherapy benefits from the IL-4 based expansion system that I alluded to earlier. And this has made our lives a lot simpler, in fact, because it means that we can make these CAR T cells from a blood draw rather than from a leukapheresis. In fact, we aim to take 120 milliliters of blood from patients, although on one occasion we only received 40 mils of blood, but nonetheless, these relatively modest volumes of blood are sufficient for the robust manufacture of a CAR T cell product containing between 2.5 and 7.5 billion CAR T cells. The right of this slide shows the percentage of transduced cells present in these products at the midpoint and on the final day of manufacture. And you can see this upward trajectory of the proportion of transduced cells over this period, reflecting the selective advantage that IL-4 confers to the transduced cells during this manufacturing process. So far, I'm pleased to say that T4 immunotherapy has proven to be safe. Following intratumoral delivery of these freshly formulated CAR T cells, we have not seen dose-limiting toxicities in any of the patients treated with doses of up to 1 billion CAR T cells. That's not to say that T4 immunotherapy is free of side effects. We commonly see that the target lesion becomes inflamed and swollen following the intratumoral delivery of these cells, and that process can persist for days or sometimes even weeks, and in some cases has been treated with systemic oral corticosteroids. But what we have not seen is any evidence of significant leakage of CAR T cells out of the tumor site and into the peripheral blood. And for this reason, we have not seen dose-limiting toxicities or indeed on-target off-tumor toxicity in these patients. One of the questions that were commonly asked is about persistence of CAR T cells in these tumors. This is quite a difficult question to address, although we do have paired tumor biopsies from a subset of the patients, and in some cases we have seen detectable signal present at a week or 2 weeks post administration of CAR T cells. From an efficacy perspective, thus far of the 16 treated patients, 10 of these patients have achieved stable disease by RECIST at 6 weeks post-treatment. I think this is encouraging because of the fact that these patients, when they receive no treatment, have rapidly progressive tumors at this phase of the disease. And as I have explained earlier, all of these patients will die of their disease within 30 weeks. This is a Kaplan-Meier curve, which is based upon intention to treat analysis and demonstrates the fact that the patients treated in this trial appear to survive for longer. The black arrow on the x-axis of this survival curve indicates the time point at which all historical controls will have died of their disease. And we did have one patient who did very well following T4 immunotherapy, a man who received 10 to the 8th CAR T cells. He achieved stable disease by RECIST and then went on to receive pembrolizumab and TVEC in a second clinical trial. He went into complete remission following this second intervention, and that remission has been durable to this day, a period of over 3 years. The other secondary endpoint that we have been exploring in this study has been the migration of the CAR T cells from the target lesion. I've already mentioned that using either qPCR or flow cytometry, we have not been able to detect any systemic leakage of CAR T cells. In one patient, we have also labeled up a very small number of the CAR T cells with indium, and we have conducted SPECT-CT imaging. And what you can see, interestingly, is that at the time of injection, or indeed 24 hours later, there is clear signal present at the target site. But by a further 24 hours later, you can see that these cells have migrated across the oral cavity to a site at which no macroscopically evident disease was present. We think this represents migration of the CAR T cells through the highly disrupted lymphatic drainage of that tumour. So at this point, I'm going to change tack and move from the clinic back into the lab to tell you about a technology which we are developing through Leucid Bio and which we refer to as a parallel CAR. This builds upon the fact that we have seen various generations of CARs developed over the years, which involve the incorporation of co-stimulatory modules to amplify the signal provided by the zeta chain within the CAR. Second generation CARs, which contain either CD28 or 4-1BB, have proven to be highly effective in the context of haematological malignancies, reflecting the importance of co-stimulation in the success of this technology. But perhaps somewhat surprisingly, third generation CARs, in which these co-stim domains are arranged linearly, have not really impacted in any kind of meaningful way over and above what we have seen with the second generation precursor. And that led us to speculate that perhaps the third generation technology is not properly enabled because the two co-stimulatory domains are not in physical proximity to the plasma membrane of the T cell. In the PCAR system, what we have done is to separate out these two co-stimulatory modules on two separate chimeric receptors. So in other words, what we are doing is co-expressing a second generation CAR alongside a chimeric co-stimulatory receptor, which contains a distinct co-stimulatory module. The first system in which we tested the PCAR approach involved chimeric receptors which were targeted against colony stimulating factor 1 receptor. And this is a rather busy slide here, but essentially what we have done here is to perform re-stimulation experiments in which we iteratively re-expose the CAR T cells to a fresh tumor monolayer which expresses CSF1R. And what we found was that with second or third generation CARs, these all behaved in more or less a similar manner, enabling re-stimulation through 2, 3, 4, sometimes 5 cycles. If we co-expressed a first generation CAR with a dual chimeric co-stimulatory receptor, placing CD28 upstream of 4-1BB, we did not see any advantage with that arrangement. However, the bottom of the slide shows 3 parallel CAR systems in which we co-express CD28 with either 4-1BB, OX40, or CD27 using this parallel configuration. And as you can see, in all cases, we can re-stimulate these T cells through more cycles. And when we move that PCAR technology in vivo in a lymphoma model, which expresses very low levels of the target antigen, we can see that while third generation CARs have no therapeutic impact in this system, the PCAR can eradicate the tumor in 3 out of 9 mice and delay tumor progression in 2 further animals. Having demonstrated that proof of concept with the CSF1R system, we then moved to the most clinically validated target antigen, which of course, in the context of CAR T, is CD19. And as we have heard from the earlier presentation by Rogier, we know that the affinity of the targeting moiety in the context of CD19 CAR-T immunotherapy appears to be important to maximize not only efficacy, but also to get the right balance from a safety perspective. So conscious of this, what we did was we took the FMC63 single-chain antibody fragment and we mutagenized that to generate a panel of mutants. We ranked these mutants in the context of a 28 zeta second generation CAR using the Z-movie technology. So the F2 CAR, shown in black here, represents a standard 28 zeta CAR in which the targeting moiety is unmodified, FMC63. And you can see the binding curve generated using the Z-movie platform indicated by the black line and indicating that the avidity of these CAR-T cells is quite high. Some of the mutants, such as mutant number 1, have no detectable binding avidity in the assay, performing similarly to untransduced T cells, whereas other mutants perform in a kind of intermediate way. And what was interesting for us to learn was that the two binders in the middle, which I have labeled as the Goldilocks binders, were the ones which ultimately generated the best CARs and the best parallel CARs when assessed from a functional perspective. This is another example of the kind of data output generated using the Z-movie system. This is a single run from a single donor in which we are plotting the dislodgement of individual cells against the force required to achieve that. And what you can see is that many of the cells are dislodged in the early phase, but you see this tail of cells which bind more tightly and which allows the discrimination between the different mutagenized single-chain antibodies which were used in this series of work. You can also plot the data as an avidity score, where the avidity of the untransduced T cells is set to 1. And once again, you see this pecking order of binding avidity of these various CARs. And another way to present the data is shown here. This plots the minimal force required to dislodge 90% of the untransduced T cells. And you can see that you dislodge relatively fewer of the CD19 specific CAR-T cells, but again, there is a ranking order which emerges in these studies. Moving that on to function, what you see here is an in vivo xenograft study using the well-established NAM6 leukemic model, where if we treat these mice with PBS, you can see rapid progression of the leukemia. If we treat with a relatively low dose of CAR-T cells, labeled as F2, where the FMC63 single-chain antibody fragment is unmodified, you see a degree of delay of progression of that leukemic burden. The in-house generated CARs are numbered on the top, 2, 4, and 5, and in each case there is a further delay in leukemic progression. And when we convert those CARs into a corresponding PCAR, again we see a further delay in disease progression, as a result of which we see a significant prolongation of survival, particularly with the PCAR engineered T cells. Here's another example of the PCAR technology targeted against the MUC1 mucin. Many years ago, we found that MUC1 was a particularly difficult target to engineer traditional linear CARs against, largely because of the steric hindrance imposed by this mucin. But what we find, if you look at the killing assays on the top of this slide, is that when you move to the PCAR system, you see a shift in the dose response curve indicated by the transition from orange over to green in these curves. On the bottom left, you see pooled restimulation assays, similar to those I described with the CSF1R system. And you can see that these PCAR T cells, on average, can be restimulated through more cycles on fresh tumor model air in the absence of cytokine support. The bottom right is a xenograft experiment. It's a triple negative breast cancer xenograft 468 cells which express MUC1, and you can see that the second generation CAR shown by orange has virtually no therapeutic activity in this model, whereas the PCAR shown by green can control this established tumor burden. Another example builds upon the T4 CAR that I alluded to in the beginning of this presentation. It was natural for us to make a P-CAR derivative, and when we compare the function of the 2G CAR or the P-CAR in vitro, we see perhaps a small advantage in favor of the P-CAR, both in terms of cytotoxic activity shown on the left and cytokine production shown on the right of this slide. When we then move this in vivo into a head and neck tumor xenograft model, what we found was that both the second generation CAR, shown in purple, and the P-CAR, shown in red, could completely eradicate this established head and neck tumor burden. But by day 63 in the tumor-free mice, we were able to conduct a tumor rechallenge, and we could now see clear blue water between the 2G CAR-treated mice, in which a smoldering tumor burden was established, and the P-CAR-treated mice, in which this tumor rechallenge was rejected. Another example showing the same principle with CARs and P-CARs targeted against the alpha V beta 6 integrin. This is a challenging xenograft model based on BXPC3 pancreatic tumor cells. They express alpha V beta 6 integrin, but with the 2G CAR, you really only make a dent in this tumor before you see rapid tumor relapse. With a P-CAR derivative, by contrast, you can eradicate the tumor in 3 out of 5 mice with delayed tumor progression in a 4th animal. NKG2D ligands are another fertile group of targets for the P-CAR system because they're very widely expressed in many solid tumors and some hematological malignancies. Again, you see a similar principle summarized in this slide, whereby these NKG2D P-CAR T cells can be re-stimulated on tumor monolayers through many more cycles than conventional CARs or control T cells. And when we test the function of these cells in a 3-dimensional spheroidal model of pancreatic cancer, in which we co-express tumor, shown in green, and tumor-associated stroma, shown in red, you can see that the P-CAR can completely disrupt those tumor spheroids, but we do not see this with any of the controls. When we move this system in vivo against that BXPC3 pancreatic tumor model, what we see is that at either a low or a high dose of these P-CAR T cells, we see tumor eradication in the majority of the mice. And what I think is telling about this model is that on the top you can see in blue and red 2 examples of T4 CAR T cell immunotherapy of this model. Even though ERB dimers are expressed by this BXPC3 xenograft, you can see that these CAR T cells have very limited anti-tumor activity, reflecting the refractory nature of this xenograft model. And in fact, we were able to re-challenge tumor-free mice in this experiment, and 4 out of 5 mice, which were re-challenged in this manner, rejected that second injection of BXPC3 tumor cells, leading in turn to a survival advantage for these treated mice. So I'll end on that note. There are many people to thank across both the clinical trial that I described and also the P-CAR technology. I'll mention Mark Davies, who played an instrumental role in the early development of T4 and also NKG2D P-CARs. I'll also mention Sophie Papa, who has been, who's a medical oncologist who has been helping me to deliver the clinical trial that I described, and also all of my colleagues at Leucid Bio who are developing the P-CAR system. Thank you all for your attention. Thanks once again to Lumix for the invitation to present today, and I look forward to receiving your questions. Thank you.
Thank you, Dr. Reijmers and Dr. Maher, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Our first question looks like it's going to be for Dr. Reijmers. Could you use other effector cells other than T cells instead?
Yeah, thank you.
It's a good question. The system is actually generated to measure interaction strength between any cell. So you can seed your target cells on the glass surface, and then come back with your secondary cells. And we have used T cells, but also NK cells, T cells with bispecifics, and also in the past we've been using B cells, for example. But it's also possible to turn it around, and then you can seed your effector cells, for example, and come in with your target cells. So yes, there's more than T cells only. Thank you.
Wonderful. Our next question, we're going to scooch on over to you, Dr. Maher. Do you know how long CAR T cells persist in the tumor after injection of T4 immunotherapy?
Yeah, thanks, Alexis. I mean, this is a question we're always asked. It's a really important question, and of course, it's a very difficult question to actually assess in practice. So I touched on some of the things that we have tried to do to begin to address this question in the presentation. So we have taken paired biopsies from a subset of patients, I think 3 patients in total. One of the challenges you face with biopsies from these kind of tumors, particularly following treatment, is that the quality of the biopsy is often somewhat compromised, perhaps because of the inflammatory response to the treatment and, of course, the propensity for sampling error, etc. But we have designed an RNA-scope probe to enable us to search for CAR T cells, and I think we have seen some evidence of persistence of CAR T cells at 1 to 2 weeks out post-treatment in some of those biopsies. I guess there are also clinical pointers to persistence of CAR T cells in these target sites for a matter of weeks, because generally the patients who receive intratumoral CAR T cells develop quite a pronounced local inflammatory reaction, which really begins within 12 to 24 hours of injection of the T cells, and in some cases this has persisted for weeks. And 1 or 2 of the patients have received corticosteroids because the reaction was uncomfortable for them, and that generally abrogates or reduces the local inflammatory reaction. But in some cases, when the steroids are stopped, we see these target sites beginning to become inflamed once again, suggesting that the cells do persist for a matter of weeks.
Great. Thank you so much for that answer. Our next question is, how do you avoid T-cell attachment onto the glass in the Z movie?
Yes, thank you for that question. It's a good and relevant question. So first of all, we have our protocols optimized to have a confluent layer of target cells on the glass surface, which usually surpasses 90%. But obviously there is some bare glass still present. So our software can detect T cells that attach to the glass, and we also block the glass surface. So we use serum for that. So the T cells, although not measured, will directly lift off when the force is induced. So they do not really bind, and the software does not recognize them. Or it does recognize them, but it excludes them from the analysis.
Great. Now moving on to our next question. Do you think there is a standard optimal avidity for CAR T cells, or is this likely to vary depending on target antigen?
That's a very interesting question from Dr. Wilding. It's very difficult to be categorical at this stage because our experience with the Z-movie technology amounts to a single target antigen and set of CARs. So I think we'll probably need to acquire more data with other antigens before being able to definitively respond to that question. However, my instinct is that it's probably going to turn out to be a case of requiring precise optimization of avidity for individual targets, given all of the complexities associated with the different targets that we look at, such as, for example, their distance, the distance of the target epitope from the tumor cell membrane, its accessibility, its propensity to post-translational modification, glycosylation, etc., etc. So I suspect it's going to be a case of optimizing for individual target antigens.
Now, does more than one CAR T cell engage a single target cell on the slide?
Yeah, that's a good question. So usually with the standard assays like cytokine release or cytotoxicity, it's very important to have the right E to T ratio to measure effector functions. With the ZIMOVI, we actually have tuned the protocol in such a way that we have low E to T ratio, but that is not very important because the measurements are at the very beginning of the efficacy of T cells, which is the binding to the target, the recognition of the target, and formation of the immune synapse. So because of that, we actually never see two CAR T cells attached to one target cell. So that and besides that, if you see that, the software package allows you to deselect these cells if you're not interested in these cells. So this gives you some freedom as well. So thank you for the question.
Great. So we do have time for a couple more questions. These are some great questions from the audience. So thank you so much for those. I do want to remind that any questions that we are unable to answer today and those that come in during our on-demand period will be answered by the speakers via the email address that you provide at the time of registration. So Dr. Maher, this next one is for you. Do you have any evidence that dual co-stimulation is delivered by the parallel CAR system?
Yes, I mean, that's an interesting question. And I guess co-stimulation by CD28 is relatively straightforward to measure at a functional level because you can look at things like interleukin-2 production, proliferation of CAR T cells, etc. And we do clearly see that with the PCAR system. Co-stimulation by the TNFRs can be a little bit more challenging to uncover, but one of the readouts that we've been interested in looking at has been NF-kappa B activation. So we have carried out some experiments with a reporter cell line, an NF-kappa B reporter cell line, and what we see is that with 4-1BB containing second generation CARs, that we get a nice signal with that reporter system as we might expect. But when we move to a third generation CAR in which CD28 is placed upstream of 4-1BB, the signal is somewhat diminished, suggesting that the functionality of the 4-1BB unit within that linear endodomain is compromised, whether it be because of the geography of the CAR, e.g. the co-stim domain is too far away from the plasma membrane, or whether it be perhaps because of steric factors whereby the two co-stim domains prevent each other from working properly. When we use the same system to evaluate PCAR, the PCAR system, we see a stronger, if anything, for NF-kappa B reporter signal than is seen even with the 4-1BB containing second generation CAR. So I would say that's the primary indicator that we have obtained of TNF-4 co-stimulation using that system. Maybe one other final comment on that subject. I mean, there have been quite a number of mechanistic studies carried out in recent years comparing co-stimulation by 4-1BB and CD28 containing second generation CARs. And it's interesting, actually, if you look at phosphoproteomic analysis, for example, it seems to me that there are more similarities than differences between the two. So it's probably quite difficult to unpick to a degree these kind of nuances in terms of the quality of co-stimulation that you're delivering. But we have carried out gene expression analysis using nanostring, and one of the hits that was pretty clear in favor of the PCAR system was a stronger cytokine signal upon initial stimulation of the PCAR with cognate antigen. So I think that's possibly another indicator that we are seeing firing of both co-stimulatory units together via the PCAR system.
Great. Thank you so much for that answer, Dr. Maher. So our next question, Does the Z-movie also work with with modular CAR T cells that engage an adapter molecule? For example, CD16 CAR T cells that bind antibodies.
That's a very nice and interesting question. So the short answer is yes. The somewhat longer answer is that Z-movie allows any type of cell-cell interaction, be it with just normal receptor ligand interaction or via a modular CAR T or with antibodies. So we also have performed experiments with a collaborator working with bispecific antibodies. And there you can also very nicely see that if you use the specific T-cell engager that acts on CD3 and its target compared to a negative control, very nicely has a very high avidity curve compared to the negative controls. So as long as you are looking at cell-cell induced interaction, the Z-movie can measure these type of interaction strengths.
So our next question, there are a couple parts to this question. So for introducing the PCARs, is it a single vector or co-transduction? And how many CARs can you put into one T cell? And the last part of it is, have you tried non-viral methods?
Okay, well, very nice series of questions there from Dr. Kloss. Regarding the first question, so we use a single vector. I mean, nowadays it's pretty straightforward to co-express 2 or 3 transgenes using ribosomal skip peptides from a single retro or lentiviral vector. Once you get beyond that point, it starts to become a little bit of a struggle, but the PCAR requires the co-expression of 2 different chimeric receptors, so that's straightforward. In some of the work we've carried out on the PCAR platform, we have also co-expressed the 4-alpha-B chimeric cytokine receptor, meaning that we have 3 transgenes that we're co-expressing, and that has also not been problematic. And the second part of the question, how many CARs can you put into one T cell? Well, I've possibly covered that in the comments that I've just made. So I would say up to 3 transgene expression of 3 transgenes using a pretty standard retro or lentiviral vector is feasible. Beyond that limit, you're beginning to struggle. Have we tried non-viral methods? I think the short answer there is no, although I would be very interested actually, because I am impressed by some of the data which is beginning to emerge using a number of non-viral methods, transposases, etc., both in terms of transgene expression, state of differentiation of T cells, and I think that's possibly going, and also cost of goods, of course, and I think that's possibly going to translate in the clinic in the not too distant future. So I would certainly be interested in trying non-viral methods, although I've been a bit of a dinosaur over the years in sticking to our retroviral system.
Wonderful. So it looks like we've got time for maybe 2 more questions for our speakers today. Our next one, is it possible to pick the interacting CAR T cells for further analysis?
Yeah, thank you for that question. This really shows that I've explained the technology well, because indeed, and also as John actually showed, each T cell that detaches from the target cell has its own interaction force. And yeah, that means that the T cell batch that you use displays a large variety in interaction strength. And perhaps there is a T cell population within your batch that can be highly interesting. And for that, of course, it would be very interesting to sort these cells. For now, we have our first generation Z-movie analyzer on the market. So this does not allow for cell sorting so far. It is a project that we have kicked off actually, and that we would like to develop in the next 2 years, which will allow you to sort the cells after they detach from the monolayer. So yeah, interesting question, and something that we are pursuing with Lumix. Thank you.
Wonderful. And it looks like we've got time for our last question, and we'll scooch back on over to Dr. Maher. What are your plans for clinical development of the PCAR system?
Well, that's, again, a very interesting question, and quite a few ideas really spring to mind. I guess the best validated target in CAR T at the moment is CD19. And I have shown you some data on the panel of CD19 PCARs that we have developed through Lucid. And so we would be very interested in evaluating a CD19 PCAR in the clinic, perhaps in the refractory lymphoma setting. So that's plan number one, I guess. Similarly, in terms of the other PCARs that I described with potential application in the context of solid tumors, I guess I'm particularly excited about the NKG2D targeted parallel CAR systems, given the fact that these ligands are a marker of stressed cells, and of course tumor cells are a form of stress cell, and the vast majority of both solid tumors and haematological malignancies express at least one of these NKG2D ligands. I think Celiad as a company have done a fantastic job in developing the prototypic NKG2D ligand targeted CAR systems, and they have several clinical trials running in the context of solid tumors and hematological malignancies. And to my way of thinking, the safety data that they have generated to date is very encouraging from the perspective of targeting NKG2D ligands. So I would be very keen to evaluate our NKG2D parallel CAR system in the context of refractory solid tumors which express these ligands.
Thank you again, Dr. Reijmers and Dr. Maher. Do you have any final comments for our audience?
No, I think we had some very interesting questions, and I would just like to thank everybody for joining our webinar, and have a good day.
And could I just take the opportunity to second that? I'd like to thank everybody for tuning in, some familiar participants and some very interesting questions. And I'd also like to express my thanks to Lumix for hosting this webinar.
Wonderful. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information that you provided at the time of registration. We would like to once again thank Dr. Reijmers and Dr. Maher for their time today and their important research. We'd also like to thank LabRoots and our sponsor Lumix for underwriting today's educational webcast. You can view the webinar on demand. LabRoots will alert you via email when it's available for replay. That's all for now. Stay healthy, stay safe. Until next time, bye-bye.